This experiment is another example of using physical agents to help control the growth of bacteria. Specifically, for the experiment, you're going to be looking at UV light. Just as a reminder, physical agents either inhibit the growth or maybe even actually destroy the growth of not just bacteria, but fungi, viruses, etc. Previously, we've talked about the effect of heat using things such as moist heat, dry heat, pressurized steam, such as what is used in an autoclave. You always have to determine whether the microorganism in the vegetative state is killed, but also are there spores there? Spores are typically harder to kill than the vegetative cell, so you have to keep that in mind. Some other examples of physical agents that we have not discussed yet are UV light, osmotic pressure, and that's what's going to be discussed here. Ultraviolet light or UV light, the way it works is that it interferes with the DNA-based sequence. It's going to disrupt some of those bonds. It's going to prevent DNA replication from occurring properly. And so it may cause mutations, or it may actually, which ultimately may kill the cell, or the damage can be so severe that it just outright kills the cell. It depends on the amount of time that the organism has been exposed to the UV light. The longer the time it is exposed, the more dangerous or detrimental effects you will see. One thing you have to keep in mind, when you are doing this experiment, you're going to grow the bacteria on a plate. You're going to inoculate the plates and then expose it to UV light. So in this experiment, and then obviously if you are using UV light as a means of controlling the growth, you have to remember a couple of things: that UV light cannot penetrate through plastic or glass. So in this experiment with the petri dishes, you're going to have to remove the lid of the petri dish during the exposure time. Otherwise, it's not going to work. UV light sometimes is used in situations where you have materials that are maybe sensitive to heat, so you can't use the heat, or maybe it's just the size. In the teaching labs, we have the goggle cases where we store the goggles that students use. After every lab, we do close and lock those cases, secure them, and then we turn a UV light on. There's a UV light on the inside, and we turn that on and run it. That is the way that we will sterilize the goggles in between uses. Some labs have UV lights typically located up along where the wall meets the ceiling, and when the last person leaves for the day, they will turn off the regular lights and flip on the UV lights and have those on all night. It will help clean the surfaces. It doesn't penetrate, say, on the tabletops, but it helps to sterilize the air that is present there. So it depends on the circumstances as to whether you can use UV light or not. Obviously, if there are people around, you don't want to cause damage to yourself and the DNA on you, so you have to be careful when and how you use it. So for this experiment anyway, you are going to inoculate plates with various bacteria. As I mentioned earlier, you will have to remove the lid during the exposure time to the UV, and you're going to have different exposure times as described in the experiment. And then you'll incubate the plates and see basically whether or not the bacteria grow or not. Drawing is just another example of a physical agent that can be used to inhibit the growth of microorganisms, because water is essential for all life, certainly as we know it. So if you dehydrate samples, this is often used like for food. If you dehydrate it, remove the water from it, that's going to prevent contamination from occurring. So that's been used for from way back, hundreds of years ago, as a means of preserving food. You do have to be careful that spores, if they are present, that those can resist the dehydration process. So you just be aware of that. Another type of a physical agent that inhibits growth has to do with osmotic pressure. You change the osmotic pressure, and that can cause cells to either rupture or swell, depending on whether you're increasing or decreasing the pressure. And in this particular experiment, one thing that you can do is you inoculate nutrient broth that has different concentrations of salt. Most microorganisms cannot tolerate salt concentrations too high because it increases the osmotic pressure. So for years that has also been used as a means of food preservation, adding salt to it, and then the food will last longer because the microorganisms can't grow in it because they can't tolerate the salt. Why not? Because it interferes with the osmotic pressure. So these are just some examples of physical agents that are used to help prevent microorganisms from growing in, whether it's food items or just growing in the lab itself.